I'm going to do something a little bit different as we talk. Here's our disclosure, since we're a publicly listed company. Today, instead of talking too much about the front of the eye, which you all know us about, we'll talk a little bit more about the back of the eye. But I just want to remind you that our lead product, Dextenza, is developed for being developed for post-surgical ocular pain, and it is a one-time insertion, delivers the entire 30-day course of medication of the steroid with a single non-invasive placement, preservative-free, and could address over 5 million surgeries in the US and over 27 million worldwide. We are in the process of resubmitting our NDA with the FDA per discussions with them. And we are in the process of expanding the label of Dextenza, including post-surgical inflammation, and that data should read out before the end of the year. Additionally, we were happy to announce just recently that OTX-TP, our drug product for glaucoma and ocular hypertension, which is a one-time non-invasive placement for up to 3 months of therapy, has entered phase 3 clinical trials, the first of the 2 phase 3 clinical trials. And this obviously has huge implications for treatment of glaucoma, since over 50% of patients don't end up taking their eye drops, and that makes glaucoma one of the leading causes of blindness. So it's not looking for new mechanisms. It's about treating patients who don't end up taking their medications and thus are susceptible. So now I want to turn our attention to the bottom part of our product pipeline, which is more on the earlier side of things and is focused on the back of the eye. These are drug depots that we are developing, and I want to talk a little bit more in detail about them. And we have a two-pronged strategy. One deals with protein-based depots, which have anti-VEGF antibodies incorporated into them, and the other is based on small molecules, or tyrosine kinase inhibitors. And the reason we have these two, I think we were going to partner here on the protein-based approach, and today, if some of you may have seen the news in the wires, that we announced a collaboration with Regeneron to co-develop an extended-release formulation of aflibercept, Eylea. And this is to be able to get what is one of the holy grails of ophthalmology, a 4 to 6 month delivery option for patients so that these frequent injections can be minimized or obviated. So we are excited to work with Regeneron. They do solid science. It's the market-leading drug. And we view that as a tremendous stamp of confidence, and we're excited to pursue this partnership. Why have we taken this two-prong approach? The protein-based antibody drugs act on the extracellular targets, and the small molecule TKIs act on the intracellular side of things. And the TKIs may have multiple targeting mechanisms, and the protein-based therapies are very specific. So I think I view them as complementary approaches. As we mentioned, we are partnering on the protein side and doing this development ourselves on the small molecule TKI side of things. So where are we with this, and what do these look like? These depots are all hydrogels, so they're completely designed to be absorbable, and they can be injected through fine needle delivery for up to 6 months of sustained delivery. So they differ slightly in their configurations. For the protein, you need to incorporate a lot more drug in, so they are designed to coil after insertion. For the tyrosine kinase inhibitors, it'll be in the form of a small rod. So I'll illustrate this in this video. On the right, you can see the protein depot. It's injected as a fine fiber, but it very quickly coils itself into a tight configuration so that it can stay out of the visual axis and remain coiled. The pictures demonstrate that this coiled depot remains coiled over a period of time, over 6 to 7 months, after which time it degrades. These can be loaded with extremely high capacity of drug, up to 50% of drug, so they don't add significantly to the volume of injection, and have been designed to be able to deliver the drug in a nice linear fashion. Some of the work we did was with bevacizumab. This just illustrates that an injection of bevacizumab will wear off after about 4 weeks, while a bevacizumab depot will continue to be active for up to 3 times as long in inhibition of vascular leakage in an animal model of VEGF-induced leakage. So we've shown this with 3 other antibody-based drugs, showing inhibition and at the same time no aggregation of the drug. For the TKIs, we have a proprietary delivery tool, which is a fine gauge injector. The fibers end up shrinking in length and expanding so that they can be very minimally space occupying. They're extremely compatible. So these are some histology. Going out to 26 weeks, you really want to see as close to 0 scores as possible over here. And I can't emphasize how important this is and how high a threshold this is for biocompatibility. When you look at tolerability, we also looked at it through OCT imaging to see fully loaded and partially loaded depots and then mostly absorbed depots and still having no impact on the retina and at the same time being active. So here is a DLA model for leakage in AMD in animals. And with an Avastin injection, you see that it's active for 2 weeks, but at 8 weeks, the effect wears off, while with a single TKI depot, the effect is sustained at 10 weeks and post that. So we've done another different model in a VEGF-induced model also. You see high sustained concentrations, up to 10,000-fold the IC50. So we're at very high drug concentration levels, and the PD effect is maintained out up to 26 weeks. So this is the first time we're disclosing up to 26-week data with a single placement. So net-net, we've made quite a bit of progress on the back of the eye efforts, and we have demonstrated for anti-VEGF protein-based depots in vivo tolerability through 26 weeks, in vitro and in vivo effect and sustained release out to 6 months. And with the tyrosine kinase inhibitors, we have demonstrated good tolerability and up to 26 weeks of activity and high sustained concentration. So we're very pleased with how this is going, and we look forward to collaboration with Regeneron. On the anterior segment side, Dextenza continues to make its way through the regulatory process, and we hope to update you shortly on that. And OTX-TP has entered phase 3 clinical trials, and we look forward to completing that and entering into the second phase 3 study. So thank you for your attention.